for delivering functional ingredients in probiotic dairy product formulation. On this topic, we are going to talk and learn about why this microencapsulation is important. There are different methods of this microencapsulation with having different release mechanism, and how it will affect by releasing that material at different rates, and then microencapsulation in different product, and our focus is on dairy ingredients, how we can incorporate functional ingredients in dairy products. So we will learn about that dairy ingredient for encapsulation, then characterizing these microencapsules, recent advancement and future opportunity, and finally we will conclude this talk. So what is microencapsulation? Microencapsulation is a technique of packing or packaging solid, liquids and gaseous material in miniature steel capsules that can release their content at control rates under specific condition. This is very much similar with having a very tiny creation of particles, which particles or those are intact in nature, and these particles they will get they will release that specific bioactive compound under specific condition. This comes under this microencapsulation. Here the substance that is to be encapsulated is usually called active capsulate or core, and the substance which is enclosing specifically that substance in the form of maybe polymer, it is usually called as shell, wall, matrix or coating material. So mostly these bioactive compounds, those are highly reactive and unstable, and there are chances of their losses of reactivity, or their concentration will get at a lesser side during the storage or during different processing which we are performing with the different products. So there are chances of having this loss in bioactivity or availability. So these microencapsulation are a better alternative for that, and here some compounds those are having strong odor or flavor which consumer will not accept later on once they are incorporated in product. And even though we want to incorporate such ingredient in product, in that condition this microencapsulation can be better alternative, like omega-3 fatty acid or fish oil. It is having like strong odor or flavor. Some consumer they usually don't accept that flavor or odor, even though we can able to incorporate such bioactive component with the help of this microencapsulation. So this microencapsulation emerges as a reliable methodology to face such type of issues and to provide further benefit as a control or targeted release, improved stability and bioavailability. Here 2-3 words are more important, that is targeted release. When these substances are need to be released, mostly they are going to get released in our gut. Unless and until that product will reach in our gut, mostly these are remain in intact manner, and with during they were traveling through our gut, at that time they will get released, and then after their effect will be on health related aspect, or that consumer will get more hello that consumer will get proper nutrition or proper amount of that specific substance. So let's see about history. Who was involved with this microencapsulation? This is Dr. Barrett K. Green. He worked on this carbonless copying paper. So in 1960s, the first studies of microencapsulation in food technology these were performed using essential oils, and scientists attempted to prevent lipid oxidation, volatile compound losses, and aroma control release. So these are main issues at that time also and present also. Our focus is mostly on to prevent that intactness or that specific characteristic of any bioactive compound. So subsequently many more studies regarding this microencapsulation of food product were published. So why mostly we perform this sorry microencapsulation? Up till now we got understood that this Microencapsulation is for protecting core material. Core material, in our case, it is bioactive compound. Then reducing and retarding the evaporation or faster rate of volatile active ingredient. We don't want that these ingredients should lose their activity. That's why we want to prevent or we want to preserve that. And for that purpose, this encapsulation will be good one. Then modification of physical characteristic in certain circumstances, mostly if you are in liquid form, some are volatile, so we have to provide them one specific condition that may be in the form of core, and within that core these bioactive compounds will be there. And this is targeted or controlled release of these ingredients, not at the not releasing whole material at the same time, but at the specific location. This is important target area. And masking taste and order, just we discussed, and enhancing visual aspect or marketing concept. So these are few justification, or on this basis this microencapsulation concept is developed. So what we have shell, we have core material, and in certain cases it is like single shell with one capsule, it is mononuclear, or single core capsule. In certain conditions it is polynuclear capsule, or when there are many capsules are intact within that shell, it is known as matrix type. So this process of microencapsulation, it is start with forming of shell, and that shell will contain that bioactive compound or core material, and finally third part is releasing that core material. In this way this process usually happens. So what are the criterias? Criterias for selection of such type of material may be based on their physicochemical characteristic, then appropriate processing condition, depending on suppose that product is processed at higher temperature and that bioactive component is not able to withstand that temperature. In that condition, this appropriate processing condition will decide which bioactive material we have to choose and which core material we have to choose. Then storage condition, and finally the storage condition of product, that is also dependent. Then physical properties, then triggering mechanism, by which means we have to release these materials so that it will provide maximum benefit to the consumer. And ultimately last important point, that is cost content. Cost contents are deciding the use of bioactive or this core material. We have to be economical when we are choosing. So all these things are trigger at the same time, and we have to choose proper combination, and on that basis we have to select both the thing, core material as well as our bioactive compound, or core material for our bioactive compound, that depends. So type of coating material, then the concentration of active ingredients, it is also important, because as per regulation or guideline, it should not be excess or it should not be under. So it may be in a proper concentration, and that should reach within that specific range and can able to give that health benefit. Again, legal issues are associated with that, and we have to choose on the basis of that the intellectual property status and freedom to use. And finally the scale up possibility with no quality concern. If everything goes well, then after we can able to produce that in a mass production, and then finally we can uh, uh, perform any trial or any claim regarding with this microencapsulated material. So there are different methods available for this microencapsulation. Among them there are numbers of methods. Scientists have studied different methods with different concentration, different bioactive compounds with their approximate payload. So that depend on how much we can able to incorporate, and finally at what will be our yield. That decide which method will be efficient or which method we have to choose. That depends on our requirement and the facility that we have. Among these many methods, this emulsification, freeze drying, liposome entrapment, these methods are quite popular, like this spray drying, then this co-acervation. These methods are good. Again, that size also matter, size of particles that we will get after this encapsulation, and finally approximate payload. So on the basis of that we have to decide which packaging material or which core material we have to choose on the basis of different factors, different parameters like this external force, pressure, within that pressure, shearing, grinding, or ultrasonics. By this way, we can release that product, or by some internal force like expansive selective permeation. In certain cases, most of the things are happen with one or two parameters like this moisture and temperature. In certain cases, when moisture content is lower down, in that condition that product or that core material is intact, but after certain condition when that product get hydrated, in that condition that material will release. Or in many cases, particularly in dairy product, that dependent on pH. pH is important one. Mostly at lesser side, these this encapsulate, they are intact in condition, but when this pH varies at that specific point, that core material will rupture and it will release our bioactive component, maybe whatever, by enzymatic action or by this pH, temperature, or by agitation, and then we will have that actual bioactive component in a where we want to release that. So that we have to decide when we want to mm, encapsulate bioactive component, and also that depend on how many or by which degree we want to release that material. So that depend on selection of method, bioactive material, as well as core material. So we will look at different encapsulated bioactive component to meal product. So mostly our target or scientists, they are focusing on this carotenoid, phenolic compound, probiotic, other bioactive components, and for that they are using different encapsulating materials like gelatin, whey protein, alginate, lecithin, others. At our college also we perform encapsulation studies, and our we use mostly most easily available encapsulating material, that is alginate. So that depend, and our we are mostly working on probiotic and incorporation of some bioactive component, and then we are converting that into microencapsules, and these encapsules we are usually adding in probiotic or fermented product. So by different encapsulation technique like ionic gelation, coacervation, liposome, spray drying, and other. Mostly in last year we tried by using two methods, emulsification and extrusion method. I will discuss about these two methods in further slides. But once that microencapsules will form, and then after we have to separate out these microencapsules, we have to dry these microencapsules, or we have to reduce down the moistures. In certain condition we have to freeze dry that microencapsule, so that their intactness, their bioactive component, they will remain in a good condition. And finally we have to, or we usually add these in functional dairy product preparation. So we can classify this into two categories: this microencapsulation addition in fermented product or in non-fermented product. So there are numbers of studies have been done regarding addition of different bioactive component like this tomato peel extract, red pepper waste extract, beta carotene extract, and they are using different method like electrospinning, freeze drying, some use the spray drying, and there are different encapsulating materials are available, and they perform to incorporate various bioactive component in yogurt type of product. So ultimately that yogurt will have these enhanced characteristics. So this type of value addition we can incorporate by using this encapsulation. So grapeseed extract, orange peel extract, hibiscus extract. So many studies have been done related with this yogurt, and by applying various techniques. Certain or in some condition vitamin D3, this is also encapsulated by whey protein or by carboxymethyl cellulose, or encapsulated in reassemble casein microcell. So this when we want to incorporate or fortified yogurt, it will give higher stability. Why? Because yogurt is already acidic product, and this vitamin D3 that will reach to the consumer in an intact form. some product are having this iron addition, like in the form of ferrous. So this iron encapsulation, and usually this, when we are adding this iron, mostly it is in the form of like ferrous sulfate. So there are many problems like that. We will get that disturbed issue, then flavor issue. So this type of problem can be solved when we encapsulate this ferrous sulfate. So it was not statistically different from the control sample. Why? Because it is in an encapsulated form. So it will not directly impart its physical or sensory characteristic. So for that condition, such type of technique is good one. Some tried like this lactobacillus acidophilus. So complex coacervation technique with peptine and casein as wall material, or some tried this ionic gelation or coacervation method, where they use peptine and whey protein concentrate as a coating material. So in certain condition like this lactobacillus rhamnosus, it is a probiotic product, a probiotic organism. This is incorporated with yogurt and added with the help of this encapsulates or microencapsulation. In cheese product also, many studies have been done, and they added various components like this encapsulation by the olive phenols, then by addition of this fennel seed extract, rosemary extract. So there are different uh, value addition is usually performed by addition of this microencapsulate, and such type of cheese, it will remain as the same as like control cheese, because these encapsule will not basically disturb that sensory property of that product. So this such type of process guaranteed the cheese constant antioxidant activity for entirely 13 days of storage period. So such type of product are of usually importance. Some like bifidobacterium bifidum addition in cheddar cheese by emulsification or internal gelation. So for that, what scientists have used, they have used beta carrageenan or sodium alginate, and under certain circumstances, the vitality of encapsulated bacteria decreases more slowly, and under stimulated gastrointestinal circumstances, encapsulate or the sodium alginate perform better than carrageenan. So there are we need to choose which one is good one, the sodium alginate or beta or kappa carrageenan. So in most of the studies, the sodium alginate will serve the purpose, and this is the most commonly used material for encapsulation. So there are many studies have been done, and scientists observe that there are more protection of probiotic organism when we compare with the control, because these probiotic organism or probiotic bacteria, they will be protected because of microencapsulation. Here we can see large microencapsulated ferrous sulfate and small microencapsulated ferrous sulfate and their impact. So this smaller size of particles or smaller size of microencapsulate, they have more recovery when there are larger type of particles. So there are different methods, and these methods giving us different particle size, but we have to choose according to our use what we want to deliver, and where when their recovery rate or their microencapsulation rate is higher, as well as yield, such type of method we have to choose. Here some collection of data related with this. In cheddar cheese, this bifidobacterium someone tried by using this emulsification method. Just we discussed this. Then Iranian UF white cheese, soft goat cheese. So there are many different work already published. Someone tried this encapsulation in kefir product also by using gelation and gum arabic by this coacervation method. In this coacervation, there is separation of two phases. Those are in a liquid form, and for that they are using some certain agent like this transglutaminase. It will be used, or it used in this particular example as a cross-linking agent, and fortified kefir was kept for 10 days at 4 degrees Celsius. So this will give more impact by addition of this fortified, and that release of that specific material, it will be protected because of that microencapsulation. Someone tried electrospraying. Electrospraying of this microencapsule or preparation of microencapsule by addition of this lactobacillus rhamnosus. They tried this electrospraying, freeze drying, and spray drying method. So there are different methods with showing different result. So whey protein isolate plus inulin, and whey protein isolate plus inulin plus this Persian gums. There are different combination. The scientists already tried, and they observed different amount or different logarithmic growth of bacteria, and on that basis we have to choose which treatment was proper. In case of this fermented donkey milk, some scientists tried with addition of this sunflower oil. So they have added this sunflower oil. They actually perform in three ways: as a plain donkey milk, sunflower oil, and fermented emulsion. So what we can see, we can able to see that cell immersion of Streptococcus thermophilus strain in emulsion and control during the control fermentation. So here we can see both the result are near around same, not having much significant difference, but this protection of this microencapsulation of any specific component, particularly with this emulsion type of condition, it is higher. We can see here at initial stages also it is higher, and it is slightly more than the control sample. Here this is showing higher antioxidant activity. That last combination, this flavor fermented milk with alginate high methoxypeptine microparticles. So this combination showed maximum antioxidant activity after 14 days of storage, while rest two combination, they showed decrease in their antioxidant activity. There are certain non-fermented products also, like ice cream. Here also scientists added many microencapsulate or bioactive component, and many research already published related uh, with such type of product. Those are non-fermented product. Here this Lactobacillus casei and Bifidobacterium lactis, it was tried with modified emulsion, and calcium alginate was used as a encapsulating material. So this such type of experiment here, what this scientist observed, this pistachio peel extract increase the melting resistance, first dripping time, antioxidant activity, and total phenolic content. This is higher when this treatment is having higher rate of this pistachio peel extract, when there is no peel extract or with gradual increase in peel extract. So it will provide maximum release or maximum amount of encapsulate of pistachio peel extract. Some tried with different organism or some phenolic compounds, some flaxseed oil, pistachio peel extract, by using different method and different type of encapsulating materials, and they observe there is increase or there is protection of that bioactive component. Ultimately their conclusion is related to this point. This encapsulation material protect that bioactive component. In case of milk, someone tried this L-ascorbic acid by spray drying and polyglycerol monostearate, and they observed this TBA value. So their observation showed that encapsulation efficiency, particularly with this ratio 5:1, this PGMS and L-ascorbic acid, it will give maximum encapsulation efficiency like 94%. So this is quite good microencapsulation efficiency. So we can say this combination is good one when we want to incorporate both these two compounds. So here combination of different bioactive components, and these like oligosaccharide keto by spray chilling by polyglycerol monostearate, and they observe particularly with these three that this astringency, bitterness, color, and other sensory property of milk, they are unaffected by the incorporation of microencapsulated keto oligosaccharides. So this study was performed up to 15 days, and after 15 days also, this rate of release of this cheto saccharide, that is quite significant. So by this way, we can say, or we can uh, plan such type of experiment. These are related with this various type of extract or vitamin C or addition of probiotic organism. In case of dried milk also, this type of microencapsulation was performed. Many scientists are performing this, and their observation is it will protect specifically lactobacilli or bifidobacteria with the reduction of certain type of pathogens or some spoilage causing of organism. Like in this study by this Rahman et al. 2019, their products supplemented with this encapsulated probiotic strain showed a greater reduction in Bacillus cereus count. So such type of formulation can be prepared, or we can add this turmeric oleoresin also by spray drying. So for what purpose? It will have higher antioxidant activity and bioavailability in that product. Particularly this experiment or this study, they perform on a rat model, and they observe that there is quite to like 4.9 fold bioavailability enhanced by such type of encapsulation. So here some examples are there, or by different scientists by using different substances with encapsulation material. Or dairy ingredient. Dairy ingredient like this milk protein, milk fat, and milk fat globule membrane. Uh, we will talk about all of them one by one. Milk fat. So there are more than 400 fatty acids and 200 triglycerides. Milk fat fraction are known. A melting point can be used for encapsulation. So there are different methods we have to follow. For what purpose? Once they are dependent on that specific melting temperature, as well as they are or they need to protect particularly from that specific pH, and for that condition, like here tocopherol was enclosed in caseinate emulsion. So milk fat fraction high in stearin and guard antioxidant against deterioration while in storage. So such type of experiment was conducted to protect this bioactive component, and here they tried to protect this enzyme substrate flavoring system. So they added this and want to prevent losses of enzyme during cheese manufacturing. Someone tried about milk fat globule membrane. So it will be in the form of liposome, and liposome will protect that from different conditions, like this specifically having property, and that property we need to use to protect our bioactive component. In case of protein, this is mostly being used at various studies, and particularly whey protein is mostly targeted by many scientists. Both these protein have been used to build numerous enclosed system, and based on their specific characteristic, how they will behave with different substance, and on that basis we have to use. Particularly these characteristics are isoelectric point, their specific formation, heat sensitivity, and their properties. Hello. Am I audible?
Yes, you are
Yes,
very much
sir.
audible.
Okay. I thought my link. Oh, anyway, so mostly this whey protein is being targeted with different following or different approaches. Mostly scientists are following to encapsulate this type of particles. So mostly among these caseins and casein micelles or whey protein are mostly they targeted And most commonly we can use this whey protein by applying a simple method to encapsulate them in the in small nanoparticles or in microparticles. And once that microencapsules were prepared, we have to test this microencapsule by uh, looking their structure, or there is proper encapsulate or not. For that, mostly the scanning electron microscopy is used, or some are using this EPR, electron resonance spectroscopy. It will based on the application of absorbance of radiance. So there are certain technique by which we can record this micrograph. Along with this isoelectric point, we have to check, and for that specific this apparatus, or we have to check that for that release purpose, when that specific microencapsule will release that material, we have to check microencapsulation efficiency as well as yield of our microencapsulate, as well as angle will decide the nature of the specific microencapsulate, whether it is hydrophilic or uh, hydrophobic in nature, and on that basis we have to decide which type of mechanism for release we have to adopt. Someone tried this ultrasound along with spray drying. For what purpose? To better encapsulate this nisin in alginate matrix, and they observe higher efficiency, around 75%. And this technique will give better result when we encapsulate such type of antibacterial substances by application of both these two. Someone tried yeast cell, yeast cell as a novel carrier to deliver such type of mic or this protein. So this biodegradable photochemical or oxidative stability, thermal stability, no effect on sensory property. Once such type of this yeast is added along with certain types of bioactive component, like they have provided different heat treatment, then enzyme hydrolysis, and then extraction of the yeast cell. These yeast cell they were tried to prepare different conjugates based on the addition of this monodextrin and protein, and they again added this ascorbic acid before spray drying, and such encapsulate they were used as a or used in different fortification product. Particles with encapsulated ascorbic acid using this SYB or this yeast based Maillard conjugates. We can see here electronic scanning photograph. So this ascorbic acid microencapsules, they are more intact and these are not disturbed, while we can see that hydrolyzed yeast cell. So there are we can observe that this encapsulation yield is higher in case of this second part, where this ascorbic acid microencapsules were prepared. So all we know, we have huge demand, particularly of value added food or the food which is having this bioactive component, and we need to uh, feed maximum population, particularly when we are talking our Indian population and particularly our condition. So there are certain types also where we can co-encapsulate specific one or two things when there is one core material and one bioactive component. In that condition, that is microencapsulation. But when we try two or three different material, in that condition it will be called as a co-microencapsulation. So particularly probiotics, prebiotic, fatty acids, or polyphenol. So in this condition we can add probiotic plus second active component. In this condition we have two specific component, or two specific like this probiotic one, and second one is active component, so that we need to fit within that core material. So such technique is known as co-encapsulation, and there are different ways by which such type of co-encapsulation are adding additional benefit. For example, we can add ascorbic acid also, we can add vitamin D also. Here is one example, like this specific cells of this Lactobacillus plantarum, it will added with this gamma aminobutyric acid, and they then encapsulated by using core material. Uh, it is in combination with dextran, inulin, and maltodextrin. It is used as a wall preparation, and such prepared encapsulate, it was finally spray dried. So, and their response is how this encapsulation actually works on the basis of this efficiency, flowability, and drying yield. So here we can see that GABA and Lactobacillus plantarum using spray drying and microencapsule obtained after spray drying, an optical image of microencapsule produced with optimized cell wall formulation. So here we can see this Lactobacillus plantarum, their survival up to 120 days. So it is in percentage. So there is quite stable their population from 100 to more than 90, so it is quite good one. Then this GABA retention, it is also almost stable in case of such type of this encapsulation, so it is showing significant of this study. So there are different commercial products available. Those are having this certain types of encapsulate, like here dietary supplement utilizing liposomal encapsulate or this encapsulation technology. How? They have added 2 or 3 different things like this vitamin C, then essential phospholipids. So this is addition of 2 or 3 ingredients by using this microencapsulation. Such products are available on e-commerce website, either for this fish oil, then probiotic drops. Here there are certain products we can able to search on Amazon or Flipkart. This is work of my student. It was in year 2022. What he did, he used cheese whey and added cheese whey by incorporation of lactic acid bacteria, Streptococcus thermophilus and Lactobacillus helveticus, and for that that team they use this microencapsulated iron, pyrophosphate, and ascorbic acid, and they flavored that product with fennel seed extract. So this product they already developed, and we got first prize during this Adroit 2022. This was one work we did initially, and in later year we did work on the development of symbiotic milk pudding using co-encapsulated probiotic culture and L-ascorbic acid. For this we use Lactobacillus helveticus. This is probiotic organism, and ascorbic acid. We perform this co-encapsulation by 2 methods, emulsification and extrusion method, and from that method this emulsion method was promising, showed good result. And finally such encapsulate we added in preparation of this symbiotic milk pudding, and for that we standardized that product, and optimum for sugar 16%, agar powder 1.5%, isabgol 0.5%, and that product showed good sensory property. This is related with, these are electronic SEM photograph, scanning electron microscope photograph, and optical microscopic view that we observe. So we can see both in this two sides, this spherical shaped microencapsule, these are from encapsulation method, and this upper it is obtained from extrusion method, and this spherical, this beads smaller in size, these are from emulsion method, and at the upper side these are not exactly intact, but these are from extrusion method. So on that basis we selected this emulsion method, and such prepare microencapsules we added in preparation of milk pudding. So such type of milk pudding we prepared by incorporation of this encapsule, and rate of addition was just 1%. 1% rate of addition of this microencapsule will give a population of more than 10⁹ CFU per ml within that product. So it is quite good, and we obtain around 12 days of shelf life, because after 15 days of shelf life it starts showing growth of fungus. So we observed good result up to 12th days of study. And from this study we can make some conclusion that this microencapsulation is useful technique, and we have to choose such type of different ingredient, active material, and by incorporating them by using different core material, and finally we can deliver in various dairy product. Application of microencapsules in food represent a very important area for opportunity for scientists, for students. And this is at present also this market is comparatively very slow. This is like many very few products are available, particularly related with the dairies. Those are able, or those are in the marketplace with having such type of microencapsules. So formulator or scientists, they have to look on various innovative approaches, techniques for creating possibility in this technique of microencapsulation. This is from my side related with this topic. I have done. I'm not able to listen, madam.
Yes, my mic was mute actually. Sir, I was in the class, so couldn't join you on the time. Now this is for the question session. Pooja Solanki and Dharmendra have raised their hands. So, Dr. Pooja, please ask your question. Dr. Pooja, do you have any questions? Dharmendra Shukla? Dr. Dharmendra is there?
Yeah, good morning. My just
Good.
curiosity is to learn. My just curiosity is to learn that nowadays nanoencapsulation has also been coming it up. So, what is better, irrespective of this, the products and the type of material to be encapsulated? So, are we shifting more and more on this nanoencapsulation, or still microencapsulation is far better, particularly
Sir,
with these microorganisms and bioactive components? And that too in the multiple, just like in the last example where so many of these dietary fibers as well as these zinc, iron and other materials to be added in fermented foods. So what would be better, microencapsulation or nano encapsulation?
sir, as per my view, microencapsulation, considering availability of bioactive components, their core material, and cost. Nanoencapsulation, it is at present also very few development have been there for recommending such type of nanoencapsules.